All right, let's just dive right in. If you're here, my guess is you've got a massive exam breathing down your neck. Don't worry, we're going to make these crazy-looking pathways finally click for you, right here, right now.
Feeling that pre-exam dread? You've been staring at these diagrams for hours. Glycolysis, gluconeogenesis, the pentose phosphate pathway. It's just a blur, right? I totally get it. It's a lot. But think of this as your emergency cram session. We are going to build a mental map that's actually clear, simple, and hopefully will stick with you when you're sitting in that exam hall.
So, first up, gluconeogenesis. Let's start with a really simple idea. What happens when you run out of sugar, but your brain is screaming for it? Well, your body doesn't just give up, it gets creative. It makes its own.
Okay, gluconeogenesis. Yeah, it's a mouthful, I know, but just break it down. You've got gluco for glucose, neo for new, and genesis for creation. So literally, creating new glucose. That's all it is. It's your body's personal backup generator. When you haven't eaten in a while or you're fasting, this thing fires up and starts making fresh glucose from other stuff that's just kind of hanging around. You know, like lactate from your muscles, amino acids from proteins, even glycerol from fats.
So why do you even need this? Well, your brain is an absolute sugar fiend. It pretty much only eats glucose. Same deal with your red blood cells. So when you're fasting, or maybe you just ran a marathon and all your sugar stores are totally empty, this pathway is a literal lifesaver. And here's the really cool part. It's also a master recycler. You know that burn you feel in your muscles when you work out hard? That's lactate buildup. Well, your liver, which is the absolute hero of this story, grabs that lactate from your blood and turns it right back into useful glucose. Pretty smart, huh?
All right, think about it like this. If breaking down sugar, that's glycolysis, is like rolling a ball downhill to get a burst of energy, then making sugar, gluconeogenesis, is trying to push that same ball all the way back up the hill. And you can't just play the movie in reverse, it's way harder.
This is a classic exam question, so listen up. Are these two pathways just the reverse of each other? Absolutely not. Think of it like a highway. Most of it is a normal two-way street. Those are the 7 shared, reversible steps. But there are 3 parts that are like super steep, one-way downhill roads. You can't just drive back up them. To go uphill, gluconeogenesis has to take 3 special bypass routes. It needs detours.
So let's look at the map here. You can see glycolysis on the left, that cascade flowing down from glucose to pyruvate, releasing energy along the way. And then, on the right, you see gluconeogenesis, that tough climb back up. See those 3 big, bold arrows on the glycolysis side that don't have a simple return arrow? Ding, ding, ding! Those are our one-way streets. Those are the irreversible steps. That is exactly where the special bypasses have to happen.
And here they are, the 3 detours you have to know. Look, for a first-year final, you don't need to memorize every single intermediate molecule, but you absolutely 100% need to know these 3 bypasses and the key enzymes they get around. Why? Because these are the control points. This is what makes the pathways different. It's the whole reason your body doesn't just waste energy by running them both at the same time and getting nowhere.
Okay, so if your body can both make sugar and burn sugar, how on earth does it decide which one to do? You can't have both going at full blast. It's all about a really elegant system of metabolic traffic control.
I want you to focus on this one little molecule, fructose 2,6-bisphosphate. If you remember one regulator, make it this one. Think of it as the master traffic light for sugar metabolism. When its levels are high, it's screaming green light for glycolysis. It hits the gas on the main enzyme, PFK1, and tells the cell, burn sugar, we need energy now. But when its levels are low, that traffic light turns red for glycolysis and green for gluconeogenesis. It's the main switch that controls the flow of traffic.
Let's see this whole system in a real-life scenario. You have a crazy hard workout. Your muscles are screaming and they're full of lactate. They just dump all that lactate into your bloodstream. Your liver sees it, grabs it, and runs it right back through gluconeogenesis to make fresh glucose. This amazing loop is called the Cori cycle. It's the ultimate recycling program. It refuels your body and keeps that nasty acid from building up.
And there's another recycling route too, called the glucose-alanine cycle. This one's really important when you're fasting for a longer period. Your muscles might have to break down a little bit of protein to get by. When they do, they release an amino acid called alanine. Again, the liver grabs it, turns it into pyruvate, and then, you guessed it, uses that to make brand new glucose. It's just another clever way your liver finds the raw materials to keep your brain happy. So far, we've only talked about glucose as an energy molecule, right? You either burn it or you make it. But glucose has a really important side hustle. It's a specialized workshop called the pentose phosphate pathway.
Now, this is the #1 thing to remember about the pentose phosphate pathway, or the PPP. It is not about making ATP. I'll say it again, its job is not to make energy. If glycolysis is the city's main power plant, the PPP is the specialty factory down the road. It siphons off a little bit of an early glycolysis product and uses it to manufacture two super important non-energy products.
And here are the two star products from that factory. First up is NADPH. Now be careful, this is not the same as the NADH you see in glycolysis. Think of the NADPH as your cell's personal bodyguard, or maybe like a can of rust-proofing spray. It protects the cell from damage by dangerous molecules called free radicals. The second product is ribose 5-phosphate. This one's simple. It's the R in DNA and RNA. It's the sugar backbone you need to build your genetic material. Without it, you can't make new cells. It's that fundamental.
So, that rust-proofing system, that NADPH shield, what happens if it breaks? Well, this isn't just some textbook what if. It connects directly to one of the most common human enzyme deficiencies on the planet.
Okay, this diagram shows us exactly how that shield works inside a red blood cell. The whole process starts with an enzyme called glucose-6-phosphate dehydrogenase, or G6PD for short. That enzyme is step 1 of the PPP. It's the machine that makes our NADPH shield. That NADPH is then used to power up another protective molecule, glutathione, which is the guy that actually goes out and neutralizes all the dangerous stuff.
So you can see what happens, right? If your G6PD enzyme is faulty, you just can't make enough NADPH. Your red blood cells have basically lost their shield, so when they run into something that causes oxidative stress, their proteins get damaged, they rust. These damaged proteins clump together into these things called Heinz bodies, and the whole red blood cell just gets destroyed. That leads to a condition called hemolytic anemia.
What's wild is that most people with this deficiency feel totally fine, until something triggers a massive wave of this oxidative stress. This is a perfect example of how your genes and your environment interact. The triggers can be certain drugs, a bad infection, or famously, eating fava beans. There are compounds in fava beans that create so much oxidative stress, they just completely overwhelm the red blood cells' weakened defenses.
Now, you might be asking, why would such a dangerous condition be so common? Well, take a look at this map. The deficiency is most common in places where malaria has been a huge problem. And it turns out having this deficiency actually gives you some protection against the malaria parasite. The parasite has a hard time surviving in those fragile red blood cells. It's a really fascinating example of an evolutionary trade-off.
Okay, take a deep breath. We have covered a ton of ground. So let's boil it all down into one final slide. This is it. This is your cheat sheet. This is the high-yield stuff you need to lock in before you walk into that exam.
Here we go. Glycolysis breaks glucose down for energy, for ATP. Gluconeogenesis builds new glucose when you're running low. And the pentose phosphate pathway, that's the side hustle, making your antioxidant shield NADPH and parts for DNA. Know their purpose, know where they happen, and know that one key enzyme or regulator for each. PFK1 for glycolysis, its opposite for GNG, and G6PD for the PPP. Burn this slide into your memory.
You did it! You made it through. See? These pathways, they're not just a bunch of random diagrams on a page. They tell a story. A story about how your body survives, how it adapts, and how it protects itself. You get the logic now. You really do. You've got this. Now go ace that exam.